The next topic in this introduction to GCSE biology is microscopy. Okay, so obviously in biology we're looking at cells like we were talking about last week. Cells, you can't see them with the naked eye, and in order to be able to see them, therefore, we need to use a microscope. But we need to understand how microscopes work, all right? And that's what we're going to talk about in this video. So let's start, just to get an idea of the scales. I know we talked a little bit about this last time, but let's do it again. So the diameter of the Earth is about 10 million meters. That's 1 with 7 zeros after it, meters. That means the Earth is about 1 million times bigger than our classroom. All right, so imagine we're sat in the biology lab at school. All right, let's think about it. You know, this is the biology lab here. I'll draw it 3D. You can imagine inside there, this is me at the front. This is the visualizer. And then all of you are sat at the tables, you know, sat working really hard like you normally do. Okay, so this is our classroom, looking at it from the outside. Now, an estimate, you know, it's not exact, it's very, very rough. We're going to say that this is about 10 meters wide. All right. Now, if that's 10 meters wide, and then this is the Earth, okay, this is not to scale. All right, now the Earth is 10 million meters wide. All right, well, a diameter of 10 million meters. Okay, so if I want to go from our classroom to the Earth, I need to multiply it by 1 million. So just think about that number for a moment now. The Earth is 1 million times bigger than the classroom. Just think about how big the classroom is and how big the Earth is. That's 1 million, right? Well, the classroom is bigger than a typical animal cell by about the same amount. Okay, so if I take here, again, not to scale. Okay, this is an animal cell. All right, that is also about 1 million times bigger. All right, so our classroom is bigger than an animal cell by the same amount that the Earth is bigger than our classroom. So if you think to yourself, in space, when they look at the Earth, can they see our classroom? No, they can't. All right, so if we're in our classroom, can we see animal cells? No, we can't. So that's why we need to use a microscope, okay? Because cells are so small, they cannot be seen with the naked eye. You can't see them by just by looking at them. Therefore, biologists use a piece of equipment called microscope and a technique called microscopy. This technique works according to the principle of magnification. So, how does magnification work? Okay, I've drawn out these two diagrams. All right, so what have I drawn out here? All right, so this is going to explain how magnification works. So let's say we're looking at this object here. All right, it's made up of 4 small squares. Okay, they might be cells. They could be anything. All right, now this is with no magnification. Okay, now these might be too small to see, but let's say that the length of each one of these small squares is, let's just for simplicity say that it is 0.1 centimeters in length. Okay, but it's too small to see. All right, so I'm going to put it under a microscope. Okay, so this is going to be this is what I see with the naked eye. So I'm just looking at it, and I see, you know, just about see them, 0.1 centimeters in length. But I want to see it bigger. I want to see it in more detail. Okay, so I'm going to add magnification. All right, so I'm going to look at it under a microscope. Okay, and when I do that, I can now see looks like this. Okay, actually it looks a little bit fuzzy. All right, so it looks like this. Okay, and now length of one of these squares is 1 centimeter. Okay, so when I was looking at it just normally with the naked eye, it was 0.1 centimeters, and now that I'm looking at it under a microscope, it is 1 centimeter. So, what have I done? Well, I've done a times 10 magnification. All right, so the image has become 10 times larger. Now, there are two words that are very important to know, okay? So this here is known as the actual length, okay? So I'm going to refer to this here as the actual length. All right, that's how big it actually is, 0.1 centimeters. All right, now I'm going to call this thing here, all right, this is called the image. And this 1 centimeter is the image length. Okay, so when we're looking at it just normally, we have the actual length, what it actually is. But then when we look at it under a microscope, we see the image length. So don't think, well, look, that's how big it really is. It's not really a centimeter, is it? No, it's really 0.1 centimeters. But in the image that I see, it's 1 centimeter because it's had a times 10 magnification. All right. So if I then, what I need to do is, because it's all fuzzy, what do I need to do? Well, I want to make it so that I see a nice, clear image. All right. Now, a common error is to think that in order to go from here to here, I need to increase magnification. Okay, that's not correct. So to go from here to here, I do not increase magnification. All I do is something called focus. All right. So I focused the image, and that makes it nice and clear. And that's going to be important in a little bit when we talk about the microscope, and also next year when you use a microscope, you need to really understand what you're doing. All right. So the other important thing is to remember that if we talk about a times 10 magnification, I've got 0.1 to 1. So let's just do a few examples. Let's do them down here. You can do them in your book if you want. Let's say I've got an actual length of 0.1 micrometers and an image length of 2.0 micrometers. What is the magnification? Well, if you want, you can pause it now and have a go at it if you think you know what the answer is. Otherwise, I'll show you. So in order to work this out, what we do is we do the image length divided by the actual length. Okay, so we can write that out as magnification equals image length divided by actual length. So if I do that, I'm going to do 2 divided by 0.1 is 20. So this is 20 times magnification. Okay. Now, when we write this 20 times, you can write it as times, then the number, or you can write it the number and then times. Okay. At the moment, we're not going to worry too much about that. All right. But you can see how this works. All right. Let's do another one. Let's say that the image length is, let's say it is 15 micrometers, okay, and it has undergone, let's say 100 times magnification. What is the actual length? All right, so you can pause the video now and have a go at that, see if you can work that out. Okay, well, using this formula up here, I can write out, well, I know that if the magnification is 100 equals my image length, which is 15, divided by my actual length. All right. Now, if I rearrange this, okay, if I multiply both sides by the actual length, okay, I get 100 times the actual length equals 15. Then I divide both sides by 100. All right, I get my actual length is 15 divided by 100, and 15 divided by 100, 15 divided by 100 is 0.15. 0.15 micrometers. Okay, very good. Okay, so there'll be some more practice of that in the questions at the end. What I'm going to do is I'm going to stop there in this video, because that's how microscopy and magnification work in general, and then in the next video we're going to talk specifically about the microscope, the different parts of the microscope, and how to use a microscope. All right, so if you want, take a little break now, you know, stretch your legs, get up, walk around a bit, maybe get a glass of water, and then crack on with the next video.